Welcome to AP Biology. This video is on translation. Translation is going to occur at the ribosome, and during translation the ribosome is going to read the sequence of messenger RNA, and based upon the sequence of codons it's going to construct a sequence of amino acids. The sequence of amino acids is going to constitute the primary structure of a polypeptide. The process of translation is going to use a second type of RNA called tRNA, or transfer RNA, and tRNA is responsible for bringing the actual amino acids over to the ribosome. If we look at the tRNA, there are two important regions. On the bottom of the tRNA we have the anticodon, and the anticodon is going to be complementary to the codons of messenger RNA. On the other side of the tRNA we're going to see the amino acid attachment site. That's going to be the part that carries the amino acid. So let's say that our codon is AUG. That's the start codon. The anticodon for AUG is going to be UAC. And on this tRNA, the amino acid that would be attached to the top is going to be methionine, MET. So these tRNAs that have the amino acid and the anticodon are what are responsible for delivering the amino acids over to the actual ribosome. The amino acids that you use come from your diet. So when you consume protein, your body breaks the protein down into its amino acids, and then the amino acids get attached onto the tRNAs. So these tRNAs are used over and over and over again. In order to attach the amino acid to the tRNA, an enzyme is used, and you see the enzyme right here. It's called aminoacyl tRNA synthetase. And this is the enzyme that uses energy from ATP to attach the amino acids that you get from your diet onto the tRNAs. The tRNAs then deliver the amino acids over to the ribosome. If you look at the structure of a ribosome, a ribosome is made up of two subunits. You have a small subunit that you see on the bottom, and you have a large subunit that you see on the top. The large subunit has three channels in it. One channel that you see right here, that's the E site or the exit site. You have the P site in the middle, and you have the A site on the right hand. During translation, we're going to see the third part, or the third type of RNA used, rRNA. When the tRNA comes together with the mRNA, the rRNA is going to hold the two together like a vice. In terms of the RNAs, it's the least important of the three. Translation is going to occur in three steps, just like transcription. It begins with just the small ribosomal subunit. The messenger RNA comes to the small subunit. The small subunit scans the messenger RNA until it finds the start codon, AUG. When the start codon is found, the tRNA with the complementary anticodon comes and binds to it. The rRNA is going to hold the two together like a vice. Once the tRNA is attached to the mRNA, then the large ribosomal subunit sits down on top of the both. You can see that the tRNA is going to be located in the P site, the middle site of the large subunit. Next, the following tRNA is going to enter the A site. This is going to be the tRNA that has the complementary anticodon to the next codon. Once the tRNA is in this site, the two amino acids are going to be bonded together. They're going to be bonded together and form a peptide bond. The amino acid is going to be cut off the original tRNA through the process of hydrolysis. This entire sequence is going to be powered by GTP. Once the amino acids have been bound together, both tRNAs and the messenger RNA are going to shift down the sites. The tRNA in the A is going to go to the P, and the one that was in the P is going to go into the E. Once in the E site, this tRNA is going to exit. This sequence is going to keep happening over and over and over again. I think about it like a conveyor belt. To see a really good animation, check out the following animation that you can find on the website. It really shows the process very well in depth. This sequence of events is going to keep happening over and over again until a tRNA enters the site. Or I should say, this sequence of events is going to keep happening until the stop codon enters the A site. Once the stop codon enters the A site, instead of a tRNA entering, a structure is going to enter called the release factor. The release factor causes the ribosome to release the polypeptide that was created, and it's going to cause the entire ribosome and the mRNA to dissociate, to break apart. Another important concept to introduce here is a concept called wobble. Wobble is a relaxation in the base pairing rules for the third base of each codon. We know that in the genetic code there's redundancy, and if we look at the redundancy in the genetic code, let's go back a little bit, you can see that the third base of a codon really doesn't matter. As long as you have CU, you're going to be coding for leucine. It doesn't matter what the third base is. Therefore, it wouldn't make sense to make 4 different tRNAs with 4 different anticodons. One tRNA is made, and that one tRNA can correspond with all 4 of these codons. They don't always fit perfectly, which is why we call it wobble. It's a relaxation in the base pairing rules. Once translation is completed, you now have a free polypeptide, forms the basis of a protein. That concludes this video.